Hi, folks. Today in our lecture in animal behavior about animal movement, we brought up triangulation and how animals can take samples of stimuli in two different locations. And so you can take a sample of a stimuli here, and you can take a sample of a stimulus over here. And in doing so, can help to locate the source of that stimulus. Now, we talked about how you can have sequential triangulation, where these two samples are taken across time. This sample is taken at one time, this other sample is taken at another time. Let's say an animal might have moved its head from one point to another. And so at the first point, it was able to take the sample here, and at the second point over here. Or these 2 points can actually be taken simultaneously at the same time if the animal has the sensory capabilities to do that. So, for example, we have two eyes. So, if you were to hold up a finger in front of your face and then wink with one eye and then the other, you'll notice that your finger moves a little bit. It's visible in both eyes, but it is moves over to, and when I'm looking at it with my right eye, it looks like it's more on the left. When I'm looking at my left eye, it looks like it's more on the right. And if I were to move my finger out farther, then I would actually notice less of that. So when my finger is really close to me, then there is a significant difference between it. And as I go farther out, then there's less of that. And in a hand wavy explanation, I said that you can use the difference in those images that are received at the two eyes to get an estimate of those objects' distance from you. And that is, in essence, what goes on during triangulation. Now, there was a great question about when I mentioned that, the triangulation requires you having these two snapshots in at different locations in space at relatively similar times. Then someone asked, well, why two? Can it be more? And I said it could be more, and that reduces the ambiguity of the estimate. But that's as far as that we could go as time would allow in the class. I wanted to elaborate on that a little bit for those that are interested in this video here today. And I'm going to do that in a real simple example here. So I just drew that we sample from two spots here and here. Now, how does triangulation work with those two spots? Well, basically, if you have a sensory organ that, like an eye or an antennae or an ear, that receives input. You then one of the things you can infer is all of the possible distances based on the strength of the input that was received. So if you know something about how strong the signal source is, so let's say, for example, you're a male moth and you know a female moth, how strong the signal that she emits, the sex pheromone she emits right at her, then that allows you to make some estimates about if you get that scent at a particular location, that allows you to make some guesses about how far away that source might be. And so, based on the strength of the signal you get, you might say, well, it's got to be in this circle around this sense organ here. So hopefully this will, let's try that again, see if I can get this to actually, so put that back, live TV here. And so normally I can get this to work. All right, I'll just do it the easy way here. So let's get rid of that. And then I'll just actually draw the shape. Sorry about that. So this here, and I'll center it a little bit, might be all of the possible locations that if I receive a signal here, I can say based on how strong that signal is, then I know that it must be that the source must be in this cone away from me. So basically, if I get a signal and it's like, based on how strong it is, that if it's really, really, really strong, then I must be very close by. So I know that it's in this distance cone. But if it's really weak, then I know that it must be farther away. So that's what I get from this one. So now I have all this ambiguity. All I know is the distance away. I don't know where in this circle it is. And that's what triangulation would like to reduce that ambiguity. So if I also get the signal, a copy of it over here, I can do that same game. And I can say, okay, so based on how strong it is over here, then it is probably in this circle here. So I'll center that a little bit better here. So now I know that if I just use the information from this second sample, then it is this distance away from this point. So it's got to be somewhere here. Well, now I've got this information plus this information, and that tells me it's got to either be here or here. It's got to be in one of these two locations. It can't be anywhere else. That's where they intersect. That's the essence of triangulation. There's still some ambiguity, though, as to which one is it? Is it here or is it here? Now, if these are my eyes and my eyes are positioned forward, then in that case, I have effectively a plane that goes through here and I'm looking in this direction, in which case it only could be this one. So in the case of my eyes with depth perception, I only need two eyes because there is one other piece of information. It's that my eyes only look forward. They're not omnidirectional. So there is no ambiguity. It's almost as if my eyes only give me a half circle to work with, and that gives me this point right here. Now, if this stimulus comes to me in a more omnidirectional way, now in order for me to figure out if it's in front of me or behind me, I might need an additional bite at the apple. So that might mean that I need to move and take another snapshot. Or if I happen to have one more ability to take a sample, let's say I've got a third eye or a third ear or something like that, and I can place it maybe over here, then um, that third ear, maybe I'll make that a different color here. So maybe I'll make it green. Well, let's say that the source actually is right here. Let's say that is the true location of the source. Well, if I've got this other sensor that is back here, then that means that its circle of ambiguity must intersect the real point over here. So it must look something like that. Now, if I take the intersection of all three circles, then I really only have one option, and that option is that the thing must be right there. So right there in the plane, if I have three bites of the apple, if I basically can get distance information from 3 points simultaneously or very close together. So if I were able to sort of take a sample here, very quickly a sample here, and very quickly a sample here, construct these three circles, then I have a way that says, aha, that's where the signal source has to be. And in this particular case, if these here were inside my head, then that would imply that the signal source is sort of inside me. But you could imagine these circles could be much larger, and so the signal source, that might be way out here, which is outside of whatever head was holding on to these things. Now, you might think, okay, so three samples, that might be all I ever need. But if you think about other things that involve the same type of math, like GPS systems and so on, then a lot of times, in order for them to function very well, you actually need more than three points of sampling the space, a greater diversity of simultaneous input or effectively simultaneous input. Why is that? Well, a lot of it has to do with the thickness of these lines. What does the thickness of those lines represent? That is how confident you are that it's exactly this distance away from your eye, from your ear, from whatever sense organ that you're using. And so really I could redo this problem not using a line, but by using my highlighter. So let's see what that looks like. So I'll go over here And I can say, well, let's say that I'm sensing from a point, say around here. And now when I get that information, say that chemical information, that olfactory information, or maybe acoustic or whatever, I know what's the strength of the source, but there's a little bit of noise because I can't quite I can't quite tell exactly how strong it is when it came in. Or maybe there's variation in the strength of the source. We mentioned back when we were talking about communication that stereotypy, that is actually, getting these sort of ritualized stereotyped signals that are very much the same over and over again really helps with communication. And that is very much true. But you're never going to get exactly the same signal every time. That sex pheromone isn't going to be emitted exactly the same every time. And there's going to be noise. There's going to be additional variation in the system. There's going to be other odors. There's going to be other wind, et cetera, et cetera. And so it may be impossible to get a perfect distance estimate, where you're like, aha, it must be exactly this circle. In reality, it might be that based on the input that you get here, all you know is it's in between two circles. It can't be any closer than this circle, and it can't be any farther than this circle, but there could be a whole lot of positions inside here, a whole bell curve of positions inside here, based on the signal that I received or the stimulus I received. So, that's why I can model that here by like saying I'll take a highlighter. So I'll highlighter with a relatively large radius, and then so we'll say, okay, it's about this circle here. And remember, what I'm trying to indicate here with the highlighter is that there's thickness here. There is uncertainty in my distance estimate. So it could be, we might say that it's no closer than this inner ring here. And we might say that it is no farther than this outer ring here. Let me see if I can do this a little faster. Yeah, right in there. But we have no idea. So it may not, it's not out there, it's not out there, but it could be anywhere inside here. Ambiguity. So what happens when we do triangulation with two sources or three or so on and so forth? Okay, well, so I'm going to drop another, sense organ somewhere over here, or another sample of the space over here. Maybe I'll make it a little closer, and I'll use a different color highlighter this time. So green. And I'm going to say I received input, and based on the input, I get a distance estimate that is something like this. Okay. So this is saying, let's try to center that a little bit more. So this is saying that it could be anywhere in this band. It's not out here. It's not out here. It's somewhere here, but I don't know exactly where. Now look what happened to that intersection. It's not a point anymore. I know that it could be in any of these two areas, this area or this area, but I have no idea where inside that area because those two uncertainties create these other shapes there. So not only do I not know is it in front of me or behind me, but even in front of me, I don't have a lot of precision. I know it might be roughly right there, but but it might be like in a 5 degree, 10 degree, cone. So if I'm here and this is in front of me, then I know that the object is somewhere in front of my nose within this cone here. But I know I know roughly the distance, but I don't know left and right, where it is. So if I reached out for it, I might miss it. So, what does adding, another view give us? And so, if I were to put another one, so this time I'll do it in the same with the same one as same idea as last time. So I'll put it back here. We'll use a different color highlighter. So it's pink one now, and let's draw. Let's say that it's actually um, that our stimulus is actually in here somewhere. Okay, so that would mean that this distance estimate should be back here. And so look at what happened there. And maybe I need to take this and move it. See if I can move this highlighter down a little bit to be more around where I put that red one. So I'm trying to center it on that. Doesn't matter too much. Point is, I have all of this uncertainty still in this single estimate, but now look at the intersection of the three highlighters. It's smaller. Look at that. So as I add more of these, then I end up reducing the uncertainty that I have on the location of the actual signal source. So if I were to add a fourth, a fifth, a sixth, it's theoretically redundant if I trust my distance estimates perfectly. But if my distance estimates have confidence intervals, if the actual distance is unknown, it's just within some 95% confidence interval, then I might need more estimates in order to combine those so that I can make those confidence intervals smaller and smaller and smaller so they start collapsing on a precise position. So that's why you might want three, you might want four, you might want five, and so on and so forth. It's to deal with the ambiguity, and the ambiguity comes, from the extra variance could come from the environment. It could come from the sender of the signal. They just are a little variable. They're not quite as stereotyped as they could be. Sometimes they speak at this volume, other times they speak at a louder volume, other times they speak at a quieter volume. That variation in volume is going to make it unclear how far away they are, because if they talk loud, then it's going to seem like, wow, they must be really close. And then if later they talk quietly, then it's going to seem like they're very far, but in the end, they're actually the same position. So all of this variation creates uncertainty in these distance estimates. And so adding more and more of these circles. Now, notice they can't be redundant information. If you put like, if I had two readings here, it might be with the nature of the noise that the second reading will help tighten this here. But just in general, putting another one here or even another one in line with this may not actually help that much. So if I would have put that other one here and create, some line this way, that may not actually reduce the uncertainty as much. Putting it back here, sort of staggering them up in a certain way, can actually end up being more beneficial at ensuring you don't just get redundant samples, you get samples that actually help reduce the uncertainty, that are most informative. So there are more informative ways to sample the space sequentially or simultaneously, and there's less informative ways. So that's getting a little out of the scope here, but I'm at least hoping that this picture helps you understand what triangulation is, why two is enough to start triangulating, why typically when we talk about like, stereopsis, so that, we talk about your binocular vision, um, that I guess theoretically is two, but there's what we often leave out, what we often forget to mention is that the beauty of vision is you only look forward. And so if you had 360 vision, then you wouldn't actually be able to tell where things were. Now, of course, I can't see behind me, and so I'm leaving out a huge space back here. But for all of the things that I can see, I don't have any ambiguity, or I have very little ambiguity between my two eyes. So that's the other nice thing about my eyes too, is that my eyes relative to my ears, each individual eye, like if I close an eye, this individual eye is actually pretty darn precise. Like I don't have a whole lot of noise in my eye relative to what I hear in my ear. If you were to ask me how far away something was just based on sound, that's a lot harder for me to do based on one eye. On top of all of that, there are these crazy things our brain can do. So, you actually can get distance information from a single eye by using the relative sizes of things that if you already know that there's certain things in view, that are in reality a certain size. You can use that to kind of calibrate the thing that you're looking at. And so there are all sorts of interesting things you can do with your vision. And what all of those things do is they make it so that this thickness gets narrower and narrower and narrower so that two eyes are probably enough if you want to sort of be able to grab something out of the air, do whatever the things it is that you do with your vision with your stereoscopic or your binocular vision or stereopsis, basically that being able to locate where things are with your stereo vision. So having better sensors and if you don't have really good sensors, having lots of them are ways in which you can use them to figure out where things are based on combining information. So two is certainly enough. Two is really good for the eyes, maybe not so good if you want really precise ability to localize things with your ears. So, if you have multiple ears, then you can do more of that. And so, what engineers can do is they can lay down a grid of microphones. And with that grid of microphones, if they have a lot of them, then if something flies overhead, they actually know exactly where it is as it's flying overhead. And two microphones may not be enough to do that very well, but you can add 16, 32, 64. And once you have this giant array of microphones, then you can localize things really precisely, even just using sound. So, yes, you can definitely add more of these things. And the math gets a little more complex when you're talking about how do you actually, when the rubber meets the road, how you actually do the math to make this work. But why it works in even in all those cases is a version of this. So if you understand this idea of reducing ambiguity through diversity, then you pretty much understand triangulation and related topics like phased array antennas and stuff like that. But that's out of the scope for animal behavior. I hope that was helpful to you. And if you have any questions in the class, then of course, feel free to reach out to us.